MedCram.com.
Welcome to another MedCram COVID-19 update. We haven't looked at Canada in a long time, and it seems as though they've avoided a lot of these recent spikes, and their daily deaths continue to be at new lows since the pandemic broke. The big news of the day, of course, is Moderna's Rna vaccine, which we'll talk about in a little bit. Before we get to that, though, I wanted to talk a little bit about thrombosis, as we were talking about before, and some new research that's come out in terms of ivermectin as well. First of all, let's talk about thrombosis real quick. And as most of you know who are regulars on MedCram, you'll know what we've talked about in terms of the hypothesis, which was that SARS-CoV-2 hits the endothelial cells, which causes a reduction in ACE2, and that causes an increase in angiotensin II, a reduction in angiotensin 1-7, which leads to reactive oxygen species in the form of superoxide in this case. That causes endothelial cell dysfunction, which causes an increase in von Willebrand's factor, leading to thrombosis. Well, it seems as though this idea and hypothesis is now being picked up, fortunately, at a number of centers around the world. Here's one, for instance, from Russia at the State University at St. Petersburg State University. A senior research associate named Anna Aksenova is reported to be advancing a hypothesis that severe course of COVID-19 may be associated with the von Willebrand factor, one of the main components of the blood coagulation system. And this article goes on to report that the replication of the virus stimulates the development of micro damage on the vessel walls. And in response to this, the body releases von Willebrand factor into the blood, trying to patch possible holes. And as a result, the risk of thrombosis increases. And as we talked about, it says here, to date, the way in which the level of von Willebrand is regulated in the blood has not yet been fully studied. However, it is known to be stored in vascular endothelial cells in special organelles where it secretes in the form of multimers. As soon as some damage to the vessel occurs, then in order to eliminate it, a cascade of blood coagulability is launched in the body in which von Willebrand factor takes an active part. They also go on to talk about how this is affected by different blood groups, which we're seeing the ABO system affecting also mortality in von Willebrand. So there may be a connection there, as we've brought up before. She goes on to say, I hypothesize that the level and activity of von Willebrand factor might be important predictors of COVID-19 morbidity and mortality, and may also itself be involved in the disease process. In order to confirm this hypothesis, it is necessary to carry out large-scale and comprehensive research into the level and activity of von Willebrand factor in people affected with SARS-CoV-2 who have mild or severe course of infection. Well, I'm so glad that people are looking at this, because I also believe that this is one potential reason why this is happening. Here's another group from Thrombosis research that will be published next month, 2020. And this article is titled, Targeting Raised von Willebrand Factor Levels and Macrophage Activation in Severe COVID-19. Consider Low-Volume Plasma Exchange and Low-Dose Steroids. And basically what they're talking about in this article is that because von Willebrand factor is such a very large protein, that you really can't get it out of the system. They say here that large size, high molecular weight von Willebrand factor multimers, which are on the order of 5,000 to 10,000 kilodaltons in size, are so big that the only way to really get them out is through something called plasma exchange. And what they found before was that plasma von Willebrand factor levels reduce after plasma exchange. So they say that potentially in these severe patients, plasma exchange may actually be beneficial in getting these multimers down and preventing thrombosis. And here's another article that was published in June of 2020 titled, Microthrombotic Complications of COVID-19 are likely due to embolism of circulating endothelial-derived ultra-large von Willebrand factor decorated platelet strings, kind of like beads on a string. And you can read the letter below. But here's the key point. Endothelial-derived ULVWFs, multimers, anchored to the endothelial surface of the vascular wall, recruit platelets and initiate microthrombogenesis within the microvasculature, leading to large microthrombi, strings composed of platelets and Again, these von Willebrand factor complexes like beads on a string structures, where platelets firmly adhere to these von Willebrand factors instead of roll on the strings. Platelets, once adhered to these strings, are rapidly activated, causing platelet aggregation and also recruit leukocytes. Remember what we talked about in our last update, that they saw quite a number of megakaryocytes associated with this. These aggregates grow until they become sufficiently large and can no longer be held onto the strings against the force of blood flow and released from endothelial cells into circulation. It appears to us that in COVID-19 microthrombotic disease, large amounts of circulating complexes of endothelial-derived ULVWFs, decorated platelet microthrombi strings, are filtered in the microvasculature as an embolism. That means that they move until they can't move anymore, or develops in the microvascular in situ, that means right where they are, causing microthrombotic occlusion. And this is what causes our hypoxemia without stiff lungs. If you want to go back and see the MedCram video where we talk about this, that was published back in early May, go to Update 67. Of course, we'll put a link in the description below. So I'm really glad that they're looking at this because I think this could be a definite possibility. However, I want to introduce another possible reason why there could be blood clots, and that is connected to ivermectin. And then, of course, we're going to talk about Moderna's RNA vaccine here in just a second. So here's a paper that has been released but not peer-reviewed, titled Ivermectin for COVID-19 Treatment, Clinical Response at Quasi-Threshold Doses via Hypothesized Alleviation of CD147-Mediated Vascular Occlusion. So this is interesting because CD147 is a protein that is on the red blood cell. Red blood cells, of course, are found in blood clots, as well as thrombin and fibrin, etc. But you may recognize CD147 as being the entry point for malaria into the red blood cell. Well, as it turns out, as we've talked about before, the CD147 protein is also considered by some a binding site for the spike protein on SARS-CoV-2. Now, of course, this doesn't really help the virus if it goes into a red blood cell because, well, there's no nucleus in a red blood cell. And if there's no nucleus, then there doesn't need to be any ribosomes, at least in mature red blood cells, because there's no protein synthesis occurring. If there's no protein synthesis occurring, the virus can't replicate. And so red blood cells are kind of a dead-end target because they're not going to be able to reproduce themselves in a red blood cell. They can only do that in a cell that has the machinery to reproduce. It would be like somebody hijacking a plane with no fuel or engines in it. It's not going to go anywhere. However, the problem may be that these virus, at least the theory goes, that the virus binds the CD147 and tends to make these cells sticky and accumulate and cause thrombosis. And that's what this article is talking about. And for that, I want to jump down here to the third paragraph. They say here the proposed catch and clump scenario for COVID-19 has a parallel in malaria. As we just talked about, CD147 is central to the infectious process of malaria into the red blood cell. But what is it that's happening in COVID-19? Well, for that, let's go to the second paragraph. And they say here, the potential for major dose response gains is evaluated based on studies indicating that ivermectin shields SARS-CoV-2 spike protein, and that this spike protein is supposed to be able to bind to CD147, but ivermectin prevents this from happening. It also potentially prevents it from binding to the ACE2 receptor. Now, because of the abundant distribution of this CD147 on red blood cells, it's not going to be able to bind to the red blood cell, and it prevents, therefore, this hypothesized catch and clump framework. So catch and clump meaning the virus catches the red blood cells by 147 and then causes this clumping to occur, which may be the basis for this thrombosis. They say here, this framework whereby virally mediated bindings of red blood cells to other red blood cells, platelets, white blood cells, and capillary walls impede the blood flow, which in turn may underlie key morbidities of COVID-19. The reason why this is important, they believe, is because ivermectin may actually prevent not only this catching and clumping, but it may also prevent infection of the virus into cells where they can cause replication. And why do they believe that? Well, this, again, is not a peer-reviewed study. And in fact, it's not even a prospective study. But in a retrospective study, and of course, retrospective studies have all sorts of problems with them and biases, but nevertheless, a retrospective study looking at ivermectin showed that in a study of 173 COVID-19 patients treated with IVM or ivermectin in 4 Florida hospitals at a dose of 200 micrograms per kilogram, yielded a 40% reduction in mortality compared to 107 controls. And that stabilization, then improvement over 1 to 2 days, frequently occurred for patients who had a rapidly deteriorating oxygen status. Now, previously in our updates, we talked about the modality of ivermectin and how it potentially inhibited the nuclear transport system of converting and transporting viral proteins into the nucleus of the cell. This is a brand new and novel theory that is suggesting that ivermectin actually interferes with the binding of the SARS-CoV-2 virus with the receptor, not only on the cells that might cause viral replication, but also on the red blood cells which don't. The importance here being, of course, with red blood cells is that they may be the basis for this clotting in patients. Now, the thing that we have to remember, of course, again, is this was a retrospective study and not a prospective study. Here is a great article in Nature that looked at ivermectin, a systematic review from antiviral effects to COVID-19 complementary regimen. So what is the use of ivermectin in COVID-19? This tries to look at the relevant data, not only in COVID-19, but other viruses as well. And it's a great reference, and of course, we'll make a reference to it in the description below. But what does it have to say about COVID-19? They say here, ivermectin, owing to its antiviral activity, may play a pivotal role in several essential biological processes, therefore could serve as a potential candidate in the treatment of different types of viruses, including COVID-19. However, clinical trials are necessary to appraise the effects of ivermectin on COVID-19 in the clinical setting, and this warrants additional investigation for probable benefits in humans in the current and future pandemics. Notice they say here that on April 10th, 2020, the FDA issued a statement concerning self-administration of ivermectin against COVID-19, referring to a recently published in vitro study on the subject. So notice what they say here, because the in vitro study on the subject looked at the dosages. And those are noted down here because the type of range in the in vitro study is in the microgram range, microgram. Here, the blood levels of ivermectin at safe therapeutic dosages in patients is really in the nanogram per milliliter range. And so this brings up the question about, can we really take the in vitro studies and make them beneficial in the in vivo state? In other words, what's going on in the test tube in vitro? Is that really what's going on in vivo? And it looks as though you have to get pretty high concentrations, at least in these in vitro studies. That may not be possible, at least in a safe range in human beings. It doesn't mean it can't happen, but it means that we need to test these results in actual patients prospectively to see whether or not it's going to work. Okay, let's talk about the big news of the day. And for that, we turn to stat news. First data for Moderna COVID-19 vaccine show it spurs an immune response. I just want to make sure everyone is reminded that I have no stock in Moderna and no financial connection to this company. So let's see what's going on. Basically, all you need to know is right here in the first 3 lines. Moderna's COVID-19 vaccine led patients to produce antibodies that can neutralize the novel coronavirus, though it caused minor side effects in many patients, according to the first published data from an early stage trial of the experimental shot. Well, where was it published? It was published in the New England Journal of Medicine. So let's take a look at that article. And here is that article titled, An mRNA Vaccine Against SARS-CoV-2, Preliminary Report. Now remember, this is important to understand that this mRNA vaccine is really unlike any other one that has been done before. Here, you're not introducing a protein into the patient so that they can make antibodies against it and have memory made against it. No, here we're introducing a messenger RNA which will go into your native cells. And that messenger RNA, much like the coronavirus, will cause the machinery of your cell to produce the protein that is a foreign protein that will cause the immunogenic response, and that will cause the antibodies. So because this was an early trial, early in terms of phase 1, phase 2, here what they're trying to do is figure out how much of this vaccine needs to be given to have an adequate immune response without having too many side effects. If it's too small, it won't have a very good immunogenic response. If it's too large, you could have some side effects. And so it's trying to find the Goldilocks, if you will, right dose so that they can now, after determining that, do a phase 3 trial where they are planning on starting that very shortly, actually, and see how that works in a much larger population, but at the appropriate dose. So as you can see here, what they did was they looked at a number of different dosages. They looked at the 25 microgram dose, the 100 or the 250 microgram dose. And in fact, there were 15 participants in each group. And basically, the bottom line of this study, and there's a lot of data in this study to go over, and we might be looking at this over the next couple of updates, but it's kind of exciting. The conclusion was that the mRNA 1273 vaccine, which is the vaccine that they're looking at, induced an anti-SARS-CoV-2 immune response in all participants, and no trial-limiting safety concerns were identified. These findings support further development of this vaccine. Now, if we look at the results here, we can see the 25 microgram group, the 100 microgram group, and the 250 microgram group. And you can see here that over the number of days on each of those that the reciprocal endpoint titers did go up. And you can take a look at the rest of these here. I don't want to get into a lot of the details because it's very complicated. Suffice to say that at the end of the day, what they noticed was that there was 2 injections that were necessary to get the response that they wanted. Here's a nice graph, though, about what I'm interested in as well, which is what were the side effects in each of these dosages, and after vaccination 1 and after vaccination 2. And here you can see things broken up pretty well, where gray is a mild symptom, blue is a moderate symptom, and this yellow is a severe symptom. So you can see here any systemic symptom, for the most part, you could see that there was a dose-dependent relationship, and it got worse after vaccination 2. In terms of arthralgias, fatigue, fever, chills, headache, myalgia, nausea, any local symptom, size of erythema or redness, or size of induration or swelling, or pain at the site, generally speaking, was fairly mild to moderate in vaccination 1 and usually worse in the higher 250 microgram dose. Whereas in vaccination 2, generally speaking, there were more side effects, and again, worse in the 250 microgram dose group. And based on these results, and also in terms of where the company wants to go, the dose that they're going to look at going forward is the 100 microgram dose. As you can read here, it says, a large phase 3 efficacy trial expected to evaluate the 100 microgram dose is anticipated to begin during the summer of 2020, and I believe that is going to be starting this month. It'll be interesting to see how long the antibody response lasts, and if things keep up the way they are, we may have a vaccine here in the early part of 2021. I think the question of safety is still there. Sometimes the true level of safety doesn't come out until you get into phase 3 trials, and indeed even in post-marketing. And of course, phase 3 is going to tell us whether or not this thing actually prevents people from getting coronavirus. Don't forget that if you want to follow other vaccines that are being developed for COVID-19, to follow at raps.org, which we'll leave a link in the description below. There they have not only the vaccine from Moderna, but also the other ones, for instance, from the University of Oxford, and also Pfizer, etc. Coming up, we're going to talk in future updates about the long haulers. These are people that seemingly have not recovered from their coronavirus infection, not just in a couple of days or weeks, but in some cases even up to months. And why is that happening? Also, we're approaching our 100th update. Might be a good time to do a little retrospective review. Don't forget to subscribe and join us over at medcram.com for other videos and information and education on other medical concepts. Thanks for joining us.